Hello, everybody. Welcome to this next webinar. And today I'm filling in for Dr. Urizzo. My name is Penny Perkins-Veazey. A quick note to make sure you stop your video because this is being recorded. And the only person we really want to see is Hidak's face. So it's my pleasure today to introduce Hidak. Oh, I know this isn't the first webinar he's done, but it's always fun to hear them. Hidak is a PhD student who's finishing up here at NC State with myself and Dr. Urizzo. And a lot of his work has been working on blueberry cultivars and what's going on both at the texture level and the physiochemical level. And this is one of the many projects that he's been working on. This one started last year looking at different aspects of sensory texture, which is a little different from just sensory. And how it relates to the texture instruments that we've been using to study blueberry firmness and softening and all the other aspects. So Hidak, welcome. Looking forward to your presentation.
All right. Thank you very much for the introduction. Hello, everyone. Thank you for having me and giving me the opportunity to share our research. My name is Hidak Oh. I'm a PhD candidate at NC State University, and I'm working with Dr. Massimo Urizzo and Dr. Penelope Perkins-Veazey. And today, I want to share some results of our research on assessing the predictability of sensorial texture descriptors using instrumental measurements in blueberry. An important goal for many breeding programs in blueberry is improving fruit texture. Texture has been recognized as an important fruit quality trait that affects the consumer's willingness to pay. It's also associated with post-harvest quality, and it's crucial for machine harvestability as well. When we think about how do you measure texture, there are a number of ways to do it. The most direct way is to do sensory tests that characterize the texture perceived by a human, either by hand feel or mouth feel. And this could be done through a consumer panel or a train panel or a breeder, etc. The other way is to use instruments that have been developed to evaluate texture. There are various instruments available that can analyze texture using penetration methods or compression methods and using different types of probes and test settings. The pros and cons of sensory versus instrumental methods are pretty obvious. Sensory test is the direct way to assess texture, but it requires more labor and time. On the other hand, the instrumental methods are relatively cheaper. They are higher throughput, and the data is more objective. But when it comes to human perception of the berry texture, the data collected from the instruments are indirect estimates of what the human sensation would be. Because of the advantages of the instrumental methods, like cost efficiency, high throughput, and objectivity, it's generally preferred to use these instruments for texture evaluation. But the relationship between the actual sensory perception of berry texture and the measurements obtained from instrumental analysis is not well established in blueberry yet. Getting a clear understanding on this relationship is very important in order to use instrumental methods as reliable estimates of the human perception. There have been a handful of studies that previously reported the relationship between sensory descriptors and instrumental parameters in blueberry, but there were some aspects of these studies that we thought we could improve in order to have a better understanding of this relationship. These studies either used only one or two instrumental parameters, which we believe can oversimplify the evaluation, or they only used a small number of genotypes or cultivars, which limits the phenotypic variation captured by the study. So in this study, we hypothesized that we could enhance the estimation of sensory texture by using a wider range of genotypes and a wider range of instrumental parameters. Measuring multiple instrumental parameters is particularly important because multiple studies have indicated that texture is likely controlled by multiple components. A single parameter is not enough to evaluate this complex trait. The next webinar, which will be in April, we'll talk about this more into depth, about the different components that are important for understanding texture at the time of harvest and after storage, and how this information can be used for selection in breeding programs. And I don't want to spoil it too much today, so I'll save this for the next webinar. So today I'm just going to be focusing on making the connection between instrumental parameters and sensory texture. So to understand the relationship between sensory perception and instrumental measurements, we harvested berries from 43 cultivars when they were fully ripe, and we conducted sensory tests with them and 2 types of instrumental tests. I'll go into more details about these tests in later slides. This is just to show you an outline of what kind of data we collected for this study. Just one thing I want to mention is that the instrumental methods that we used here were developed for the VacCAP project to evaluate various attributes of fruit texture. The methods were designed to derive 17 parameters from the flat probe penetration test and 17 from the double compression method, also known as the TPA method. And these methods are available for sharing if anyone wants to use them. So just reach out to me or Penny or Massimo if you're interested, and we can share the methods that we used. The first part of this study was the sensory analysis. Sensory texture was evaluated among 43 cultivars using 9 sensory descriptors by 5 highly trained assessors from Sensory Spectrum. There were multiple training sessions before the actual evaluation where the descriptors were defined and the evaluation protocol was established, including what kind of external references to use and where the external references would fall on in each intensity scale for each descriptor, and what kind of technique to use to assess each descriptor. Before we go into the data, these are the definitions of the descriptors that we used, and I want to give you a quick overview of them so that they're at least somewhat familiar to you when we look at the data. There were 3 sets of descriptors that differ in when and how they were evaluated. The first was at the surface level, where springiness was assessed. So springiness was evaluated before the skin break, just by compressing the berry with the teeth a little bit and assessing how well the samples return to their original form. The second set of descriptors was assessed during the first bite or the first chew. Hardness was how much force was needed to compress the whole berry. Snap crisp was the force to break the skin, and it includes the popping sensation or the crisp feeling. And force to grind was the force required to completely bite through the sample, including the skin or whatever was in between the molars. And the rest were based on the texture while you chew down the sample. Juiciness is the amount of juice released. Dissolvability is how fast the samples disappeared in the mouth. Amount of particles is how many solid particles were perceived in the flesh. Mealiness was the dry, sawdust-like character. And residual skin was the amount of skin remaining after fully chewing the sample. Using these descriptors, we were able to characterize the sensory texture profile of the 43 cultivars. The spider chart here on the right is illustrating the average sensory texture of the 43 cultivars. So this gives us a good idea on what our sample set looks like as a whole. With this, what we can do is we can use this as a reference when we look into each cultivar and compare them with this average. So this is looking at each of the 43 cultivars individually. The gray area in each plot is the average of the scores for each descriptor. So it's what we saw in the previous slide. And so this gray area is the same for all the cultivars. The red lines are the individual texture profiles of each cultivar. So you can see that the sensory profile is different for each plot or each cultivar. And when you compare the red line to the gray area, you can get a sense of the texture of that specific cultivar compared to the average. We can see that there's quite some variation among cultivars, especially more so in some descriptors. We can see that some cultivars that stand out, for example, the so-called crisp cultivars, like blue crisp, indigo crisp, or key crisp, had noticeably higher values of springiness, hardness, and snap crisp compared to the average. We had a few that had high scores in juiciness and dissolvability, like Robinson and Vernon. Mealiness was low in all cultivars except for Titan, which had an exceptionally mealy texture, which is what we described as the sawdust-like sensation. We found out that some descriptors vary a lot among cultivars, and some descriptors not so much. For example, there's quite some variation in springiness, hardness, and snap crisp and juiciness, but there's not as much variation in force to grind or residual skin. We looked at the variation using PCA as well, principal component analysis. We see a nice spread across all the 4 quadrants. PC1 explained 43% of the variation, and PC2 explained 17%. And to get a better sense of what this PCA is telling us, I marked some cultivars that are known to be crisp or firm, like Key Crisp, Indigo Crisp, Blue Crisp, Reveille, and I circled them in blue. And some cultivars that are known to be soft, such as Robinson, Jewel, San Joaquin, and Croatan, I circled them in red. And we can see that the sensorial firmness, like how firm they are, kind of spans along the principal component 1, PC1. And if we look at the variables or descriptors that contribute to PC1, it's primarily hardness, springiness, and snap crisp. This means that these 3 descriptors, hardness, springiness, and snap crisp, may be the most important descriptors to differentiate the crisp or firm versus soft perception when chewing down the berries. But this doesn't necessarily mean that the other descriptors are less important. There was still large variation in some of the other descriptors. For example, juiciness and dissolvability had wide ranges of variability, which means that when you think about what affects the overall texture liking of consumers, these could also be important factors. Something else that we've noticed here is that the amount of particles was quite different in the rabbit eye cultivars, which are the green data points here. In fact, rabbit eye had a different range of scores in a lot of these descriptors. I don't have the significances shown here, but the rabbit eye cultivars were significantly different from the northern highbush or the southern highbush cultivars in most of these descriptors, which implies that rabbit eye blueberries may have a distinct texture profile from the other types. We tried to see which descriptor may play the largest role in making the rabbit eye's sensory profile so distinct. We looked at this using PLSDA, which is a supervised classification analysis. How this works is we basically let it know which data points are high bush and which data points are rabbit eye, and we tell it to do its best to distinguish the categories. And from the results, we can see how well it did at discriminating the blueberry types and which descriptor contributes the most to do this. And the resulting plot here is telling us that the amount of particles and mealiness are important descriptors that contribute to the difference between rabbit eye and high bush cultivars. So the next question was, are these sensory descriptors related to any of the instrumental parameters that we can measure using very sophisticated instruments like the texture analyzer? So to figure this out, we measured flat probe penetration parameters and TPA parameters at the same time that we did the sensory tests. 17 parameters were evaluated from the flat probe test and 17 parameters from the TPA test. First, we looked at the correlations. The first matrix is correlations between the flat probe parameters and the sensory descriptors. And the second matrix is between the TPA parameters and the sensory descriptors. The three key sensory descriptors that explain the firmness sensation, springiness, hardness, and snap crisp, were highly correlated with quite a few of the instrumental parameters. The purple box here is the correlations between the three descriptors and the flat probe parameters, and the green box is the correlations between the three descriptors and the TPA parameters. And you can see that a lot of the correlations were higher than 0.6 or 0.7. The instrumental parameter that correlated the most with these descriptors, out of the flat probe parameters, the closest was FM, which is the maximum force. And if you're wondering what FM or maximum force means, it's a very simple parameter you can get from an instrumental method. So this is an example of a force deformation curve you would get from a flat probe penetration test. The x-axis is the time or the distance, and the y-axis is the force. So it's the resistance that the probe is perceiving while penetrating the berry. And it's recorded over time or distance, like the graph that you see here. The force increases at first as the probe pushes into the berry, and the maximum force, or FM, is typically at the point when the skin breaks. And out of the 17 different parameters that we derived from this graph, FM correlated the strongest with these three descriptors that mostly explain the firmness variation. This was also evident in PCA results. We ran PCA with all the 9 sensory descriptors and the 17 flat probe parameters. And the 3 descriptors, springiness, hardness, and snap crisp, grouped closely with FM in the PCA results. When we look at the TPA, out of the TPA parameters, gumminess had the highest correlation with these 3 descriptors. Although there were some other TPA parameters that also had a similar level of high correlation with these descriptors. And we also confirmed this with the PCA as well, using the 9 sensory descriptors and the 17 TPA parameters. Gumminess grouped closely with these 3 descriptors here as well. If we look at some other descriptors too, real quick, the descriptor force to grind was slightly less correlated with the instrumental parameters than the first 3 descriptors, but it was still on a decent level. Dissolvability had similar level of correlations, but in a negative way. So the direction was the opposite. And the other descriptors like juiciness, amount of particles, mealiness, and residual skin didn't have any noticeable correlations with any of the instrumental parameters. Now the next question was, okay, we know that the sensory descriptors and instrumental parameters are correlated in certain ways. Can we predict the sensorial texture using the instrumental parameters without actually doing sensory tests? Because sensory tests can be quite expensive in terms of time and labor. So using the instrumental parameters that we have, I used different methods to build prediction models that can predict sensory texture. And we have 17 parameters from the flat probe and 17 from the TPA. And I use those to develop prediction models and see if we can predict the sensory descriptors using those instrumental parameters. Just to briefly introduce the prediction methods, the first approach was selecting variables to build the models using different criteria. We have all these variables or parameters that we evaluated from the flat probe and TPA. And out of those, we only leave a few variables that are most useful to build prediction models. It could be only one variable or a few more. The point of this approach is to see which parameters or variables are the most crucial to predict sensory texture. The other approaches, PLSR, lasso, and ridge regression, utilize as much of the raw information as possible. So instead of eliminating most of the variables, you leave most of the variables or all of the variables, and you're basically giving them different weights to kind of resolve the collinearity that these variables have. And because these approaches use a lot of the raw information, in many cases, the prediction accuracy goes up compared to when you eliminate most of the variables. The downside, though, is that it could be hard to interpret which variable is the most important. And also, if you want to use the prediction model built from these approaches, you need to measure all of these variables again. But again, the hope is that you'll get a higher prediction accuracy. So this is showing you an overview of the results that we got from the predictions. I know there's a lot going on, but I'll walk you through what these mean and highlight some of the important findings from this. So what I'm showing you here is the accuracy of the prediction models, where we are trying to predict each sensory descriptor. So you can see the 9 sensory descriptors here. And we're trying to predict them using either the flat probe parameters, which are the red bars, or the TPA parameters, which are the blue bars. The y-axis is the prediction accuracy in terms of R². So the higher the R², the more accurate the prediction model was. And the x-axis is the different prediction methods, like variable selection, VS, PLSR, lasso, or ridge regression. So starting off with the 3 descriptors that explain the firmness sensation, springiness, hardness, and snap crisp, the predictability was relatively high compared to the other descriptors. The R² values were around 0.6, and there wasn't too much of a difference between the instrumental parameters that were used for prediction and the methods that were used for model selection. So it didn't matter much whether you used the flat probe parameters versus the TPA parameters, and it also didn't make that big of a difference which selection method you used. And the variable selection methods gave us some idea on which parameters were the most important predictors for the selective method. And not surprisingly, FM and gumminess were identified as the key predictors to be included in the models to predict springiness, hardness, and snap crisp. For the other descriptors, the predictability was less consistent across the instrumental methods or the prediction methods. Some interesting things we found here was that juiciness was able to be predicted with high accuracy only with the TPA parameters. and it didn't work when only a few parameters were used. You had to utilize most of the variance we had among the 17 TPA parameters. So juiciness is not a descriptor that can be predicted with just one or two instrumental parameters, at least from a flat probe or TPA method. But using all the TPA parameters we measured, you can get a pretty good estimation of juiciness. On the other hand, it was the opposite for residual skin. Only the flat probe parameters were able to estimate residual skin on a decent level. And this had to be using all the flat probe parameters as well, and not just one or two parameters. And the TPA parameters here were pretty much useless to predict residual skin in our study. So overall, this indicates that some predictors could be estimated with one instrumental parameter or just a few, while for some descriptors, the type of instrumental method you use matters, and you need to utilize more than just a few parameters. Some parameters, it was hard to say that they were really predictable on a reasonable level. For example, especially mealiness, it wasn't predictable at all with any of the methods. It could be because we didn't find much variability of mealiness across cultivars, or it could be because the instrumental parameters or methods that we used weren't suitable to estimate mealiness. But for a lot of the other sensor descriptors, the predictability was at least on a moderate level. So the prediction accuracies weren't super high, but I want to point out that the models were still very useful to select for the highest or lowest values in most of these descriptors. These are the plots of observed values versus predicted values for the best model that was developed for each descriptor. The highest observed values are in blue, and the lowest observed values are in red. And you can see that, that for a lot of these descriptors, the models predicted the highest and lowest rated cultivars pretty well. These are just a couple of examples from the previous slide to show you in a larger size. On the left is the model predicting hardness using the flat probe parameters as predictors and lasso for model selection. And on the right is the model for juiciness that use TPA parameters for predictors and PLSR for model selection. And the predictions here weren't perfect. Like if they were perfect, all the dots would be on this dashed line. But you can still see that they could be used to select for or select against the extreme values on each end. So overall, we think that using multiple instrumental parameters can help better estimate the sensory texture of blueberries and also help breeding programs to select for desired or undesired genotypes for different sensory properties. To wrap up, these are some summary points. We profiled the sensory texture of a large set of cultivars with 9 descriptors and a highly trained panel. Springiness, hardness, and snap crisp were the 3 key descriptors to discern the firmness perception. FM, or maximum force, and gumminess were the instrumental parameters that were most closely related to these descriptors. And our study determined that using various instrumental methods or parameters can enhance the estimation of sensory texture. So with that, I want to thank everyone for being here and having me here. I want to thank my PhD advisors, Dr. Massimo Iorizzo and Dr. Penny Perkins-Veazey. Everyone in both labs have helped me tremendously. Sensory Spectrum helped us with the sensory test. Dr. Suzanne Johanningsmeyer, Dr. Marcelo Molinari, Dr. Lara Giongo, and Dr. Ann Pilato all helped us design the experiment and interpret the results. The Castle Hayden Research Station provided the blueberry material, and Mark and Randy helped us set up the instrumental analysis. And I also want to acknowledge NIFA for funding the VacCAP project. And before we take questions, I want to let you know that we're going to have a webinar in April on the 22nd, which will be focused more on the post-harvest side of blueberry texture and the application in breeding. The presenters will be Dr. Massimo Iorizzo and Dr. Lara Giongo. So I hope we see you there as well. Thank you very much.
Sorry, I thought I was unmuted. I'm opening thank you so much, Hidek. This was a lot of great information to see. Opening up the floor for questions.
Hi, Hidek. This is Jessica. Great presentation. I have two questions for you. So the first question is about the juiciness. I was wondering why you think the individual measures on the machines don't explain much variation, but in the overall model, you get a high explained variation.
And that's a good question. I don't have a firm answer for that. One thing that we hypothesized is that juiciness is kind of related to, how much juice or water is in the berry. It's related to the moisture level in the berry. So it could be hard to estimate it using just one simple instrumental parameter. It could be, it might be something that we would have to estimate using different parameters that are independent or discrete. And our data is kind of telling us that, it's true that juiciness is just a parameter, a descriptor that's really hard to relate to one simple instrumental parameter. And it's very interesting to see that using multiple different independent parameters can do a better job at predicting juiciness. And we saw this in different fruit as well. I remember a study in apple where they estimated juiciness using multiple TPA parameters.
Nice, thank you. I was thinking it kind of reminds me of, the reason we use genomic prediction instead of markers for certain traits. And then my second question was, on that last PCA, I think it looked like juicy and crisp were inversely correlated. Is that the case? And if so, why do you think that is?
Is it juiciness and what did you say, sorry?
I think it was juicy and crisp looked like they were inversely related on the x-axis. I guess this table also, there was a PCA plot where they were presented opposite each other in one of your last slides.
PCA, let me see.
This one right here.
Right. Juiciness and these descriptors you're saying, right?
Yeah.
Yeah, I mean, they are in the opposite direction, but juiciness really didn't contribute that much to PC1. Like in PC1 in particular, juiciness didn't really have much of a contribution to either. So it's kind of hard to say the relationship between juiciness and the other parameters from here. Juiciness was a parameter that contributed the most to PC3. So I think a more direct way to see this is, looking at the correlations. And I mean, they were negatively correlated, but they weren't significant. The correlations were very weak.
Okay. Great.
Yeah.
Thank you so much. Great presentation.
Hi, Helg. This is Juan. Very nice presentation. I just have a quick question. Do you mention at the beginning that the instruments are a high throughput method to evaluate the texture? So I was wondering what speed do you use in your tests and how long does it take for you to run all of these tests with the instrument?
That's a good question. I mean, the high throughput was, a comparison with the sensory test mainly. There are instrument methods that we used are still relatively low throughput compared to the other instrumental methods that are out there. We were mainly, since this is our kind of an early take on looking at this relationship, we wanted to use the most sophisticated and controlled instrument that could, give us a ton of information as much as possible. So it's relatively low throughput compared to the others. I can't remember the test settings right now off the top of my head. I'm happy to share that with you after this. Yeah, I'm trying to think how many berries we processed during the hour. We did the calculation recently, but I can't quite remember. I don't want to give you a wrong number, a misleading number. So I think I should get back to you on that after this. I can give an email or something.
So how many berries do you use per each test for TPA and for the puncture test?
So we used 10 berries to get 1 rep, and we had 3 reps. So 30 berries in total for each cultivar for TPA and 30 berries for flat probe.
Okay, thank you.
Any other questions out there?
On a note, Ido, thank you so much for the great presentation. I want to go back to the, to the question about the throughput. It is true that the texture analyzer it's not like most high throughput, but we have multiple data now. The next web is more about the texture in blueberry. It's not. It's multiple component contribute to texture, and we have done some correlation between the most traditional method, high throughput, and the texture analyzer, and the parameters that are correlating with those texture with those parameters. They are not necessarily the one that are explaining sensorial texture, or neither the one that explain change during the storage. So I think there are ways to speed up the texture analyzer. You can customize, the plate for running multiple sample at a time. The other thing is we have been evaluating 1,000 sample at each breeding program for the project. So it is low, but gave you the information that seems that it's needed to start to look at texture in blueberry.
I have a question.
Yes.
Hi, so Hidak, very nice, really nice presentation, very good job. So, unfortunately, the two, to me, I mean, no, it's not the two most important, but two very important parameters for consumers or for quality of blueberries, the juiciness, which is really a good, I mean, positive for a consumer perspective, and the mealiness, which is a negative instrument. So you could not really find, well, at least the juiciness, just you had really a strong strong predictor with the TPA, but the mealiness, nothing. And so did you discuss that with your team or with the sensory people? Yeah, the mealiness, nothing. It's terrible. Any suggestion that, you could find another way to measure it? I don't know, without being too complicated, like electron microscopy or to look at the cells. I mean, any suggestions from the group here? It would be nice.
Right. I mean, I don't, have other suggestions how to do it. I mean, we'd have to, try different things. All we know is that our methods didn't work from using our cultivars. And one thing that I want to reiterate is that in our cultivars, in the 43 cultivars that we measured, except for Titan, all the other cultivars had very marginal differences in mealiness. So we didn't really see a big variation at all in mealiness. So that could be a reason that we're not able to, make any models for the prediction. Yeah, not sure. I mean, it doesn't necessarily mean that these methods aren't capable of predicting mealiness. It's hard to say because the variability was so low in the set of cultivars that we had.
Right, you didn't have a full range of mealiness.
Not really,
in our
no.
samples. Yeah, okay, that explains. Thank you.
Yeah. Yeah, if anyone else has seen, a way to do this or any suggestions, that would be interesting.
And look, I want to just add another note. So we have so one of the reasons why we did this study was not originally planned, but one of the reasons we did it because we have preliminary data from the VacCAP on consumer preference versus these parameters and texture. And what we see is that with consumer preference data, there is no correlation with the parameters that we used, and largely because in the consumer panel, the consumer is not asked to evaluate specific descriptors. It's just it's good to know and the intensity, but without any descriptors. So the data that Hiduk shows illustrate that it's really important to do a panel with trained panelists. Otherwise, we will not be able to put any predictions.
Okay, has everybody had a chance to ask questions? If so, we'll wrap this up. And thank you all for participating. This has been very helpful, both from what Hidek has provided and what the participants have added.
Thank you very much, everyone. Just, give us an email if you want to use the methods that we have, or if you have any questions, we're happy to share that with you. Thank you so much.
And there's a survey coming your way, everybody. Great. Thanks very much.